having me. I'm very active in science still, even 10 years after the prize. And I'm going to speak about the latest developments in the lab that are currently pushing the scale of resolution and to the molecular scale, actually, of fluorescence microscopy. And I will show that this will have major implications for understanding the dynamics of molecules. So I will start out with a very simple question, which is how to find out the position of a fluorescence molecule with maximum accuracy, precision, and speed in the focal plane of a microscope. Now, this is a very simple question, and in fact, for about 5 or 6 decades, people had a very simple solution, a very simple answer, which I'm going to explain to you now. Usually when people wanted to find out the position of a molecule within the focal plane of a microscope, now let's assume that this green shaded area is actually the focal plane of a normal, let's say, microscope. What they usually do is that they illuminate a whole field of view and hence also illuminate the molecule that is at an unknown position. And then the molecule fluoresces and produces fluorescent light, and the fluorescent light is then projected onto a camera. Now, the camera, of course, renders an image, and what we will get is, of course, an image of the focal plane with the molecule at some point. But because light is diffracted, and so fluorescence light is diffracted as well, the molecule will not look like a point, like a little star, as it is shown here, but it will show up as a blob of light that has a comparatively large size. And that comparatively large size is actually directly proportional to the wavelengths. So the longer the wavelengths, the bigger the spot, and therefore, of course, you can say, okay, well, how do we know where the molecule is because the spot is so big? But that's not a problem really. The narrative is we can easily find out where the molecule is because the position of the molecule must coincide actually with the maximum of this diffraction blob. So in other words, once we have identified the position of this diffraction, the maximum of this diffraction blob, then we know where the molecule is because that must correlate in space actually with the actual position of a molecule in the sample. So the whole point of localizing a molecule is then to find out the position of the diffraction maximum with maximum precision, because we want to know with very, very high precision, of course, also accuracy, where the molecule is. And then if you calculate this position, and do the simple math, you will find that it not only scales with the wavelengths, but with the square root of the number of detected photons as well in the denominator. Meaning that if this fluorescence spot consists of many, many photons, then the number n is large. And of course, also the square root of n is large. And then, of course, the position is found out with higher precision because this number here gets smaller. So then we know where the molecule is, and then we can, on the camera, and then we can find out where it is in the sample. Now, this concept has been used actually for, I would say, 6 decades, roughly, in many, many biophysical applications. But since it's based on the maximization, of course, of the number of fluorescent photons, so the maximization of n, of course, it has problems that come with that maximization of the number. Namely, of course, fluorescent molecules, they tend to bleach. After a while, bleaching means they do not emit anymore, so they fade. Not only they bleach, they also blink. They go off for a while, and then they come back. And of course, during the dark period, it's not really possible to find out where the molecule is. And that's a big problem if the molecule moves. Because if it moves, of course, it may go somewhere, and then you don't know where it has gone, simply because you've not seen it. And then, of course, inherently, there is the problem that if you need large numbers n, the method becomes slow because you need to wait until you find out a certain position. And that can be prohibitively long, of course, because you need maybe 10,000 photons to get a precision of the order of a nanometer and below, and you might not get them because the molecule doesn't emit that many photons. And then there's another problem, which is very serious, actually. Molecules are not dots like this. They are very tiny. They are just about 1 nanometer in size, which is very tiny. But when they absorb or emit light, they behave like dipoles, meaning that if the molecule is oriented like this, then the emission goes always in this direction or that direction, but never in the direction which is along the axis of the molecule. And that kind of leads to a shift of the pattern in position, meaning that while we may locate the maximum of this fluorescent spot at a certain location, the actual position of the molecule is shifted to the side, yeah, because the pattern is actually not where it should be because of the orientation of the molecule. And that can easily amount to 5 nanometers, 10 nanometers, 20 nanometers. And then while you may get a very high precision, so a precision of about a nanometer in the number that you get, it may be the wrong number because the molecule actually is shifted to the side. Now, these problems are really prevalent actually with this type of method. Nevertheless, this method is being used now in thousands of labs for tracking the position of molecules. It is used in the super resolution methods called Palm and Storm and Paint and so on. So it's a very common method, but the limits are very obvious. So my lab worked actually on an alternative way of finding out the position of the molecule, which you will see in a moment will give higher precision, better accuracy. So we will know with much higher confidence where the molecule is and at higher speed. And I'm going to explain to you now what the basic rationale behind this idea is of localizing the molecule with a different method. Namely, let's assume now we have the same molecule sitting in the focal plane of a microscope, and we want to find out where the molecule is. Now, rather than illuminating the whole field of view, we do something else. We decide to create a coordinate in sample space with the light that is emitted from our light source, which is a laser usually. Now, and that can be done by converting a normal laser beam into a donut-shaped beam. So that's why you see this kind of ring-shaped, donut-shaped pattern of light. So that pattern of light is diffraction limited, of course, so it has a size of a diffraction limited beam, but the position here of the minimum is perfectly defined. Why? Because it's made of many photons, and you can easily find out where there is the point where a photon is never showing up from the laser. So that's the minimum, the point where because of the physics of diffraction, a photon is never ever going to show up. And this point is extremely well defined. It can be defined to, say, sub-angstrom, picometer, whatever precision. And then, of course, we know always where this point is, because not only is very well defined, but we can also push it around in the sample space with a beam deflector. So we have a kind of mirror or something that tilts its position, or some other device, and the result of that is that we can push this donut around in sample space at will with arbitrary precision and with maximum accuracy. And then the whole localization process, finding out the position of the molecule, boils down to finding out where the molecule is with respect to the known position of the donut zero. So since we know actually where the donut zero is here, where this minimum is, once we know where the molecule is in relation to that donut zero, we know where the molecule is in total, of course, in absolute terms, within the focal plane. And so the question is now, why is this a useful concept? Simply because it's relatively easy to find out where the molecule is with respect to the donut zero, because if the molecule is here, for example, then of course there is excitation light, because you see there's green light that would make the molecule emit light. But when they coincide in space, then there is no excitation going on. There's no fluorescence going on. And then, of course, the molecule remains silent, although it's there. Yeah, so it remains silent. So we can tell actually from the brightness of the molecule where it is in relation to the donut zero, and that's the strength of the concept. Now, please note there is no camera here at the detector. There's just a normal detector, because we need only to count the emitted fluorescent photons. We don't need to do anything else. And as I said, if the molecule is slightly off, there will be some excitation going on and some emission going on, and hence some measurement, of course, of fluorescent photons, but only weak. But if it's, of course, very far away like this, then it will be strong. So in other words, the brightness tells us how good the match between the two is. And once the match is done, we know where the molecule is. That's very simple. Now, why is this concept superior to the concept that you've seen before in all aspects? In order to make this clear, well, I've come up with a thought experiment. With my last name being Hell, I decided to introduce a demon. And this demon, of course, kind of does things that we humans cannot do, because demon is very clever. And let's imagine for the moment that if the molecule moves, the demon has the capability of just shifting the donut zero on top of the molecule with no time, within no time. So within an attosecond, if you like. Attoseconds have become very fashionable lately. So imagine that in no time, the demon puts the donut zero on top of the molecule. Now, if that is the case, yeah, then the molecule will never ever emit a fluorescent photon. So the detector will never make a click or something. Yet the molecule can be perfectly traced with arbitrary precision by the demon. Why? Because the demon knows, oh, there is no emission going on. So the place where I put the donut zero must be the place of the molecule. So the demon can trace all the time the molecule without requiring any single fluorescent photon. Now compare this with what you first initially. So initially we had a concept that need to maximize the number of fluorescent photons. But here we have a concept that doesn't require any. Yeah, so it's the total opposite. And clearly this concept has a major inherent advantage. So the advantage is that we can precisely localize, at least in this conceptual limit of the existence of a demon, with no fluorescence emission. The other striking consequence is there is no wavelength dependence on the localization. Remember that in the previous case there was this lambda on the top of the equation to the wavelengths, but here the wavelength doesn't really matter because we are just matching 2 points, the donut zero and the molecule. It doesn't really matter if the donut is made of green light, of blue light, yellow light, X-rays, microwaves, whatever. It's only the point zero that matters. Now, also the dipole orientation is gone for the same reason. Now, this is actually a thought experiment. There is no demon. It's clear that these conditions cannot be totally fulfilled in practice. There's no way. But what we could do, we can engineer the method such that we come as close as possible in reality to this ideal. And that means, in fact, that we replace the demon with a controller with an electronics. And we have a detector in here that counts molecules. And then the controller checks actually how many, not molecules, photons, how many photons have been counted, and then acts on the beam deflector, and then pushes the donut zero around such that it matches as good as possible the position of the molecule. So if the molecule moves, there will be detections, of course, because the superposition will not be perfect. But based on those counts, actually, the donut zero can be pushed around, and then we can locate the molecule. Now, this method requires much fewer fluorescent photons than the initial method that you've heard before, that has been the one that has been used for decades. Why? Because it kind of comes closer to that ideal that requires no emission. So we save many fluorescent photons that we need to get a certain precision. And because of that, we are faster, because we don't need so many photons, we don't have to wait so long. And then the molecular dipole orientation also is also ameliorated by far. And if you go down through the mathematics, then you find that the precision, of course, scales now again with the square root of number of detected photons. That has not changed. That's part of the Poisson statistics, but it doesn't matter. But what has changed is that there is not the wavelengths here, wavelength lambda, that plays a role. The wavelength cannot be changed. Once you've used a certain laser, it's fixed. But this distance L, and this distance L is, so to speak, the average distance between the molecule and the donut zero, meaning that if we manage to reduce this distance on average, then if you reduce this, then the precision sigma goes up because the number L, of course, goes down, then sigma goes down, meaning that precision is higher, because higher precision means smaller numbers of sigma. And this is very good because now we have a handle on increasing the precision. Previously, we couldn't do it because the wavelength was fixed. This relies on the maximization of photons. But now, if we manage to get these two as close as possible, precision goes up. Now, at this point of development, we can make L so small that we require 100 times fewer detected photons than with the camera. So we can be 100 times faster or 10 times more precise. Yeah. So that's very useful because with a certain number of photons, we are 100 times faster or 10 times more precise. Now, 100 times faster is very interesting for a given precision because this allows us to address, say, very important questions in biology, such as the movement of motor proteins. Like this is one of the most famous scientific cartoons, the movement of kinesin on a microtubule, and kinesin carries cargo like neurotransmitters and so on. And there have been a lot of studies, pioneering studies, very good studies, of course, showing that the molecule works in this hand-over-hand scheme. They were all done with a camera, so with the old way of localizing molecules. That's why the method were relatively slow. So in order to get a precision of 1 nanometer, it took the experimentalists at that time 350 milliseconds. So it's quite a long time because camera recording is slow. And this was done by actually attaching a fluorophore to this stock. Since we are faster, 50 times, 100 times faster, we can redo the experiments and check whether we can discover new things. So I'm not going to dwell too much on the most recent, say, findings, but I would like to show you that this movement, which is again shown here as a cartoon, was recorded with our method. And we could see for the first time, because we had this high precision substeps, like this ones, you know, instead of 8 nanometers, 4 nanometer substeps. And we could measure them with the fluorophore, which was not possible before, simply because the precision was not there and the speed was not there. Also, we could use full physiological concentrations of ATP to let the motor run at its native speed. That was not possible before, so they had to slow down the motor and so on. I'm not going to go into the depths of the study because you're not interested in the detail. But I'm just going to allude to the fact that, of course, you get new insights. We also looked at dyning, of course, endogenous dyning that was labeled with a new method directly in a living neuron, in an axon. And here we could see so-called pauses for a long time. People think there is a kind of tug of war between dynein and kinesin to change directions because dynein transports just in the opposite direction from the periphery to the nucleus. We've seen that very rarely, but we've seen a quick reversal, which was not seen before. But the point I would like to make here, this is for the first time that dynein stepping was ever seen in a living neuron directly with minimal invasion. So it shows that the method really adds to the repertoire of studying motor proteins. And if I let's assume I was 30 years younger or something, then I would probably go into this field because many things can be discovered now due to the fact that there is a fundamentally new tool at hand. So not only we can be 100 times faster, we are also 10 times more precise. And this really makes a difference because the precision becomes now so good that we can really look at distances between molecules that are really at the molecular scale. So let's imagine now we have two fluorophores, yeah, the two stars, and they are at distances that are very close, below 10 nanometers. Now, so what people do so far to measure such small distances, they resort to a method called FRACT. That's an energy transfer between two fluorophores. I'm not going to speak about that, but it's heavily used in thousands of labs throughout the world, because there's no other way of measuring distances optically directly in fluorophores. So because our precision now becomes so good, we can say we can measure them directly. And so we can measure with super resolution the distance between two fluorophores that are that close, say below 10 nanometers. Now, you should know that the basic enabling element of all super resolution methods that exist today, no matter what they call it, STED, Palm, Storm, Pain, whatever, yeah, they rely on a so-called on-off principle, meaning that if you have two molecules that are very close, you make sure that only one of them emits, because otherwise the signal of the two gets confounded, yeah? But because if only one is emitting, then you see only the signal from that molecule, you localize that molecule, find out where it is, and then you know where it is, and then you stop it, then you turn it off, and you turn on the next one, localize this one. So this on-off principle is really key to all the methods, no matter what they call it. As I said, STED, Palmstone, whatever. What we do here in the next experiment, we really measure distances between two molecules that are very close and turn them on off individually. So when this one is localized, the one on the right-hand side is off and vice versa. Now, in order to prove that we can see such small distances, we have prepared a ladder of prolines that changes in length. These are proline chains. If you have 5, then they're very close. If you have 20, they're much further apart. If it's 40, then it's even much further apart. So we can vary the distance roughly between 3 and 11 nanometers. In a not very controlled way, because they do some cis-trans isolation, but we can get examples where they're very close, like here, 2.8 nanometer, and examples where they're 7.8 and 9.5 or 11.4. And this is within this Fred range. Yet the measurement here shows that we can really tell them apart, distance 2.8 nanometers, precision 3 angstrom. So we have an optical method, MINFLUX is an optical method that really allows us to see distances at this scale with very, very high precision. And this is, of course, a new tool because you don't have to measure distance indirectly through a method like FRET or so, which is very dodgy and has its problems. You can measure directly by imaging. And the distances are really crazy. So even if you have molecules that are so close, like 11 angstrom, 1.1 nanometer, as it is shown here in this dimer, then you can locate them and measure the distance optically, see, okay, it's indeed 11 angstrom. So how do we know it's 11 angstrom? Because it's known from NMR. So a completely different method, autonomial method. And can we get any closer than 11 angstrom, 1 nanometer roughly? The answer is not really, because the fluorophore itself has this size of 1 nanometer. So it's impossible to get closer. Of course, you can get apparent distances in projection. Yeah, if they are still 1 nanometer apart, but the projection is, of course, smaller, then you can get smaller numbers, but the Euclidean distance stays the same. And so here we have smaller numbers like 3 angstrom, even 1 angstrom. But if you verify the 3D distance, it's still 1 nanometer. So basically, the message here is we're down to nothing. Resolution has become so good in super resolution fluorescence microscopy that you really are down to basically fluorophores touching each other. And this is kind of, has it been expected? Well, you can say yes and no. When I started the field back in the early 90s, of course, I had high hopes. I was young. I was 30, even younger than 30. Of course, I had high hopes to one day get to the molecular scale, but it was totally insane at the time. Really, believe me, it was really insane. And if I said that, people would have said, this guy is, sorry, he's sick. But anyway, it's here, and you should not underestimate human ingenuity. And also, once you have found the fundamental principle, you can work on that principle and constant engineering, and so improves it to a level that is not imaginable. And I think we will see this phenomenon many times in the 21st century. We've seen it many times in the 20th century, in fact, and we will see it many times in the 24th century, that development will take us to where you could not have ever imagined that it would be possible to take us. So, and there's another big surprise coming up. This is very recent. It just came up basically experimentally this year. As I said, for separating all these methods Stead, palm, stone, pane, the ones that got the Nobel Prize, all rely on this on-off principle, turn molecules on-off sequentially. But that's a fundamental problem in the concept, because if you need to see the fluorophores sequentially, you cannot see them at the same time, of course, inherently not. If you want to track both at the same time, it's not possible, because you have to turn this off and then it may go somewhere, whereas the other one you can track, of course, but this will go somewhere. Then you turn this one off and then this one may go somewhere. And that's a problem if you want to follow both at the same time. So I've been questioning myself, can I beat this fundamental tenet actually of the whole field of super resolution microscopy? If there's a way of beating that, it would be a big step, of course. And we figured out, and I realized that in the end, it's possible. And I would like to explain this to you under certain conditions, that this can be done. This is something very new. It's coming out in a couple of weeks in Nature Physics, but I'm going to revisit, of course, this famous diffraction barrier again. So why did people say that unless you turn on off, of course, two say simultaneously emitting fluorophores are not separable? Let's assume we have now two fluorophores that emit at the same time, and we have this normally focused Gaussian beam that kind of scans across them, and what we'll get is, of course, a beam that looks like a Gaussian because they are so close. If you had one, it would not be a big difference. If you had two sitting in the same place and the same, this patch, of course, would look like this patch. You can hardly distinguish it. In fact, you could make a subtraction to see if there are subtle distance as a result of this distance between the molecules. There would be something, but it would be completely within the noise band, because you have a finite number of fluorescence counts here, say 1,000, for example, and then it's always in the noise band, because you don't get exactly 1,000. You may get, in one case, 850, in the other case, 1,000 or 81 or something, and then it's always in the noise band. And that's why people said it's not possible to distinguish in a confocal microscope, for example, molecules that are much below the diffraction band. That's why you need to turn on off and do a super resolution method like Stead or Palm or Stone. Okay, but let's do the thing we've done just a few minutes ago. Let's scan with a donut. Let's assume we have a beam that excites molecules, has a donut zero, and the molecules sit in the same place. What would you get? If they sit in the same place, the image, so to speak, of the two fluorophores would be a donut. It would look like this, yeah? Zero here at the center, zero fluorescence, but stronger fluorescence here at the donut crest, because if the zero coincides with the molecule, there will be no emission here. But what happens if the two molecules are slightly further apart? If they're slightly further apart, there won't be a zero in here. Why? Because it's impossible to get a zero in here. Because if the zero coincides with that molecule, the other molecule will emit, and vice versa. And if the zero is in the center, both of them will emit. So the zero here will not be a zero. And if you subtract the two, there will be clearly a measurable difference. And this difference is measurable because we measure against zero. Yeah, we can measure against zero background. So in other words, this distance or difference actually that we measure between the two is beyond the noise band, as you can see here. So it can be distinguished from noise. And hence, in other words, now we can distinguish the two molecules from those where they sit in the same place. So here with the donut, if we scan with the donut, we can separate, whereas here if we scan with Gaussian, we can't. And so in other words, we can overcome the diffraction barrier, even though the molecules are emitting at the same time and constantly on. And even if they are closer, it becomes even easier, as a matter of fact, because the absolute noise band goes down. So this is conceptually very appealing. And here is the proof. Here the molecules were 35 down to about 8 nanometers in distance. Still, 8, 9 nanometers, they could be distinguished. So this is something that they published in arXiv already almost a year ago. Meanwhile, it's even better than that. But this is kind of unexpected physically, that you can really distinguish molecules at this distance, even though you keep them on all the time. And why is this interesting for biology? Because now we can track two moving molecules at the same time, yeah, without having to force them going on off, yeah. So we can. People said, now if you want to track two molecules, you have to have a red one and a green one. We don't have to have that. They can be of the same kind. And this allows us and not only two, also you can have three or four, as it is shown in here, to come up with a vision, which I think is very, very appealing. There are many methods now by which you can get a structure of a protein, like X-ray, crystallography, NMR, cryo-EM, and of course there's AlphaFold, which got a Nobel this year, but there's not many good methods to see how proteins actually work mechanistically. But now we can come up with a concept where you have fluorophores sitting at critical sites of the protein, and then if we make that protein do some work, they will change the position. And we should be able to see the change in position of the molecules and hence infer actually on the positional change of the different protein sites. And so this is a vision. It's not realized yet, but I think it'll come because it's physically sound. 3 years ago, 4 years ago, you would have said, or I would have said, there's no way of doing it because this principle was not discovered at that time. But now it's clear and it's sound, and it should tell us how proteins work. So in other words, where super resolution kind of told us where proteins are in a cell, this method should tell us how they work mechanistically. So there's a lot of options that are coming up now. And since I'm coming to the end, I just want to make a little, say, kind of pun here. So MINFLUX compares actually to the old type of localization, like electric vehicle to the combustion engine. Combustion engine, you need to scale up the number of cylinders in order to have a high torque. And this Bugatti, which is a very fancy car, very expensive, it's 16 cylinders. Normal car has 4 cylinders, but this one has 16, but doesn't scale really. You see, it scales very badly. 4 times or 8 times the cylinders doesn't really make it much better. If you just get rid of this concept and have a different concept, 0 cylinders, different engine, you can have high torque and higher speed accordingly. And so here you have a sluggish function. In fact, with MINFLUX, the precision scales up exponentially with the number of the total photons detected. I'm not going into details of the mathematics here, but this, the gist of it is a different concept because different concept you can break boundaries. So with that, I'm coming to the end. I don't have big group as you might think. I'm at 2 places, that's right, but I have very few and carefully selected people who work with me. And they have contributed, of course, a lot to this development. There is no doubt about that. And we've having a lot of fun pushing this development. And I'm thanking them, of course, and I'm also thanking you for listening so patiently. Thank you.
Thank you, Professor Hell, for the lecture. Right now, we have Professor Liu to moderate the Q&A session. Professor Liu, please.
Okay, good morning, Professor Hell. Can you hear me?
I can hear very well.
Thank you for the fascinating talk. This is the most beautiful and most powerful donut I ever see. It reveals the secret of life. Okay, good afternoon, everyone. Welcome to Singapore and welcome to GYSS. The floor is now open for questions, but maybe allow me to ask the first question to Professor Stefan Hell. So yes. You received your PhD degree in physics in 1990. And then somehow you get a Nobel Prize in chemistry. And then you went to the German doing the biological research and dealing with a lot of electronics, right? So what's your major and how do you pick up those things over the years?
Yeah, I have a very comparative education in physics. I studied physics like, say, it's a very general, everything, with nuclear physics and everything. And I specialized very late, actually, during my PhD. It has big advantages and disadvantages. I mean, the advantage is that you have a very general knowledge of the field, and it forces you to get very deep insights into how nature really works. I mean, of course, how physics really works. And physics, of course, was attractive to me because there is many interesting underlying principles and patterns. And this has helped me later on because, so the way I looked into this super resolution field was very much from the physics perspective. So I want to understand what makes it tick? What is the key? Like I realized it's the on-off principle. And then of course, once you realize that, then you know, okay, which is actually the Achilles heel of the concept, and what can you do in order to push it further? People who specialize very early, like they specialize in biotechnology or they specialize in I don't know, optics or something. Usually, the tendency is that they are not so much trained conceptually, but they are trained in knowing a certain field very well technically. And that becomes an engineering thing. And it has many advantages because then you can make many advancements that are technical or incremental, maybe also conceptual. I'm not saying that. But a narrow specialization at a very young age may kind of limit your capability of getting the broad view and getting and making perhaps a deeper transition in a certain field.
I see. Thank you for
That's
sharing. Yeah, for young people, don't
And,
get.
and just to answer your question now, did I learn biology? Formally, not really. I mean, I just spoke with people and I learned by talking to them. And chemistry, the same thing. So I have no formal training in chemistry. Actually, I had some chemistry my first semester when I studied physics, but then I didn't do any serious chemistry afterwards, but learned it by talking to people and, of course, getting trained by reading or so.
Thank you for sharing. So don't get your specialization too early, and we keep learning from the peers around us. Okay, the floor is open. Please, your question, please.
Hello, I'm Bahar. I'm from University of Turku, Finland, and I have one question about that slide that you showed that like you kind of transitioned to donut-shaped imagery that kind of showed the differences in the peaks. I was just wondering like how did you what was the logic behind this transition to this donut-shaped scannings? I just don't know how you like go from the first, form to the second form. Like, what causes it?
I see. And then I can answer your question technically. Technically, it's not very difficult to make donuts nowadays. What you do is actually there is a piece of glass that has a certain shape, if you will, that causes a phase delay if the light passes through it. And the phase delay is such that that different parts of the wavefront, so if the wavefront comes from a laser, are out of phase with respect to each other. So if this part on the left is out of phase, the 180 degrees out of phase from the other part, then the light from here that interferes with this one will go dark here, and the same here. And so you can make opposing parts out of phase, and then you will have a zero in the center. That's not a big deal. You can buy that and put it into the beam, and then you get that. So making donuts has been around for ages. I didn't invent that. But I realized that useful for microscopy.
Okay, thank you.
Thank you.
Thank you. Are there any other questions? Oh, there is a, yeah, please.
Hello, Prof. So my question is like at young age, when you were not specialized in the field, how did you go against a scientific norm? What kept you motivated and believe in that you are going in a right direction?
Yeah, it's a good question, and I alluded to that already. So when I actually got this idea when I was a PhD student, and I was not tasked with looking into the diffraction barrier. I was asked to back computer chips with normal laser scanning microscopes. And I want to do something profound because I was trained as a physicist with a very general view. And of course, I had to study basic principles. And then doing my work as a physicist in that very technical surrounding, I had to give up all the principles, and didn't think about principles anymore because I was forced to think very technically, and that frustrated me enormously. And so that's why I thought about maybe there is still something fundamental to be done with light microscope. And this was very outlandish at that time because everyone was convinced that there's no way of doing something with a diffracted beam. You cannot get high resolution because of this diffraction. This is known, and so then I figured out that a possible way to go about it is to play with the states of the fluorophore, because I realized that people have just not used state transitions in fluorophores to overcome the diffraction barrier. They use them for different things, measuring pH or measuring maybe fret distances or something, but not for resolution, because resolution was considered a closed subject. And then when I opened the field and did that and went out first and said, I think it's possible to overcome diffraction, there was big misbelief, and many people actually put many obstacles in my way. So why did I believe it? Because, as I said, I was trained as a physicist to think about concept, and I realized the concept is correct. I was sure the basic physics behind it is right, and that made me go on. So I felt that problems that are left are only technical. The technical problems can be sorted out. New technology. There's new technology coming up basically every year. You see it in cell phones and everything. New technology is always coming up. If the concept is correct, then it will work out. And that kept me going. So if you have a good idea and you know that the concept is right, and there are maybe some shortcomings, technical shortcomings, you can kind of bet on the fact that it'll work out if the goal is desirable, because the technical problems are likely to be solved over time.
Thank you. Allow me to abuse my power a bit to allow last quick question, please.
Yes, thank you for the great talk. This is like somewhat technical, but I was interested in like, what's the kind of algorithms that's used to sort of like trying to find the sort of like a matching, and this can have like profound impact on the speed of this microscopy, right?
Yes. So what actually happens is we quickly scan the donut to a number of positions, or quickly, continuously around, count the presence of emitted photons or the absence of emitted photons, and then based on those numbers, we figure out where the molecule is most likely going to happen. And based on that assessment, we reduce again the scanning error, so that L is continuously reduced by making an assessment. And there is, of course, there's a trade-off between how long do you wait to have a decent assessment and how many photons you need to collect, and that depends on the background. So if you have little background means there is counts that come from the fluorophore. If there's little background, you go very quick and you get very high precision. The technical limit in the end is the background.
Thank you.
Okay, thank you, Professor Hell, for the fascinating talk. And I'm looking forward to see you in person. And thank you, everyone.
I will come to Singapore at some point.
Yeah, sure. Looking forward. And thanks, everyone, for your attention. Thank you.